Hello, a warm welcome to you in the Histology of Respiratory System class. The competencies expected are: you should know the histological features of various parts of the respiratory system, the nasal cavity, it has a respiratory part and olfactory part. Epiglottis has a lingual surface and a laryngeal surface. The larynx has true and false vocal cords. You have to know the histology of the trachea, bronchus, bronchiole, lungs, and related applied anatomy. Before going through the details of the histology of the lungs, I want to show you the picture of the normal lungs. See in the left panel the appearance of the normal lungs. It has some pink areas and white areas. The white areas are the alveoli. But see, in the COVID infected lung, what happened? There is very little white areas. That means alveoli are full of this pinkish material. And what happens? This alveoli is for exchange of gases. So, if the alveoli is full of exudates and inflammatory material, there will not be exchange of gases, and a person will suffer from respiratory problem. So I have taken it from a very recent literature, although postmortem studies or pathological studies in the COVID patients are very little. So there is intra-alveolar fibrin deposition and patchy areas of organization in COVID infected patients. This picture shows the higher magnification of the alveoli of a normal person. See, the alveoli has valves and interalveolar septum, and alveoli is looking like a triangular structure or oval structure, pentagonal structure. Inside the alveoli, normally air is there. But here, in the COVID infected lung, what happens? The whole of the alveoli is full of exudates, inflammatory material. There is hyaline membrane formation, fibrin plugs, and interstitial inflammation as evidenced by the number of inflammatory cells. So, to understand the pathology of the lung or the diseases of the lung, we have to understand the normal anatomy of the lung. The functions of the respiratory tract. First is air conduction. The second function is air filtration. Filtration starts from the nasal cavity. In the anterior part of the nose, there are vibrissae or some hairs. Those hairs entangle the coarse particles, coarse dust particles, more than 10 micron in diameter. Less than 10 micron diameter dust are dissolved in the mucus secreted by the different cells of the respiratory tract, and less than 2 micron dust particles directly goes to the lung alveoli. The next function is air exchange. Exchange of oxygen and carbon dioxide occur in the alveoli. Voice production. The larynx is voice box. There are vocal folds. The vibration of the vocal fold modulates the voice and produces legible language or sounds. Smell. The posterior one-fourth or the upper one-fourth of the nasal cavity contains alveolar contains the olfactory cells, which is for the smell. The neuroendocrine cells are there in the whole of the respiratory tract, which secretes some hormones and immune responses are there for the whole of the mucosa because it is a part of the BALT or bronchial associated lymphatic tissue which has immune function. Parts of the respiratory system. It has conducting part and respiratory part. Conducting part means it conducts the air, and respiratory part means where the respiration occurs. That means exchange of gas occurs. In the conducting part, there is no exchange of gas. The conducting part consists of the nasal cavity, which has smaller divisions like vestibule, the anterior part is called vestibule, the respiratory area, and the olfactory area. Here, the black mark area is the olfactory area. Then, paranasal sinuses are there, the nasopharynx, larynx, trachea, bronchi, and bronchioles. So after this is the conducting part. That is a part of the bronchiole where the alveolar sacs open. So exchange of gas is possible in that part. So that is called respiratory bronchiole. So the respiratory part starts from the respiratory bronchiole. Then the smaller subdivisions are alveolar duct, alveolar sacs, and the alveoli. The details of the nasal cavity. The nasal cavity has anterior part which is lined by skin. This part is also called vestibule, and here there are lots of hairs or vibrissae are there. Already we have told the function of the vibrissae is to filter the coarse dust particles more than 10 micrometer size. Next to it is the nasal mucosa, where there are three elevated areas. Those are called the corners. They increase the surface area of the nasal mucosa, and there are lots of blood vessels in this mucosa. You might have experienced blockage of the nose during cold infection. What happens? There is dilatation of the blood vessels due to bacterial antigen or viral antigen, which which causes inflammation of these areas where the mast cells come and they degranulate and they release certain substances like histamine. So they cause vasodilatation and fluid comes out through the capillaries and it produces edema. So that is how during common cold you get nasal obstruction. Normally also in physiological state you also see sometimes what happens, one of your nasal cavity is blocked, and this happens alternately. This is just to protect the receptors of the olfactory mucosa so that the receptors get rest for recovery. Then after 15 or 20 minutes the other nasal cavity opens up. The function of this mucosa is to cool the air, because normally during this time, during the summer time, uh, you are inhaling hot air. This hot air, if it goes directly to the cavities, then it will dry the mucosa and kill the cells. So the function of this nasal fauna are to cool the air, inhaled air. Behind the nasal cavity is the nasopharynx. Nasopharynx, there are tonsils also in this area. So any inflammation of this area causes tonsillitis or inflammation of the tonsil. Some of you might have experienced secretion from the nose. This happens due to inflammation of these areas, also inflammation of the tonsil, the lingual tonsil, pharyngeal tonsil, palatine tonsil, etc. So air enters through the nasal cavity, it comes into the nasopharynx, then it goes to the oropharynx and laryngopharynx. The part of the pharynx behind the nasal cavity is called the nasopharynx. Part behind the oral cavity is called the oropharynx, and part of the pharynx behind the larynx is called laryngopharynx. Air passes from the nose to the nasopharynx, oropharynx, then it goes to the larynx. The opening of the larynx is covered, or there is a lid-like structure which is called epiglottis. We will see the structure of the epiglottis later. So the nasal cavity has a vestibule which is lined by stratified squamous keratinized epithelium which has vibrissae, and it has sebaceous glands also. It has a respiratory area. Respiratory area is lined by pseudostratified ciliated columnar epithelium, and there are 5 cell types: the ciliated cells, goblet cells, brush cells, small granule cells, and basal cells. The whole respiratory tract is mainly lined by these 5 types of cells. You have to remember the main types of cells are ciliated cells, goblet cells, brush cells, small granule cells, and basal cells. The olfactory area is the upper 1/4 of the nasal cavity. It is much thicker than the respiratory epithelium. It is also lined by pseudostratified epithelium, and 4 types of cells are there. The main types are the olfactory receptor cells, there are supporting cells, there are brush cells, and basal cells. The respiratory part of the nasal mucosa, there is a large part of the nasal cavity, and it is lined by pseudostratified ciliated columnar epithelium. It has columnar cell, goblet cell, brush cell, small granule cells, and basal cells. This area most exposed to the air, epithelium is higher and has more goblet cells. There are 3 conchae. The function of the conchae are they increase the surface area in contact with the air and air turbulence to allow more efficient conditioning of the inspired air. It has a lamina propria also, which is highly vascular, as already mentioned, that because of this vascularity, there is inflammation and lot of fluid comes out, which causes edema of the choana. Therefore, during common cold or any infection of the nose gets frequently blocked. There are 3 major paranasal sinuses and small sinuses are there. The maxillary sinus is the largest one, followed by the frontal and the groups of ethmoidal air sinuses. These air sinuses are lined by pseudostratified ciliated columnar epithelium, and they drain into the nasal cavity. The function of these air sinuses are air conditioning function. They just cools the inspired air. The larynx. This is the anterior view of the larynx. Larynx contains some certain cartilages joined by ligaments. These are epiglottis, the thyroid cartilage, and the cricoid cartilage, and some small cartilages are there. Inside the larynx lies the two folds, the vestibular fold and the vocal fold. The upper part of the larynx is formed by the epiglottis, which is lined by respiratory epithelium. The laryngeal part is lined by the respiratory epithelium, whereas the lingual part, which comes in contact with the food material, is lined by stratified squamous epithelium. In between the vestibular and vocal folds, there is cavity which is called sinus of the larynx, which contains lot of mucous glands. The function of this mucous gland is to moisten the vocal folds. The vocal folds do not have any gland. The part below the vocal fold of the larynx is also lined by respiratory epithelium, that is pseudostratified ciliated columnar epithelium. Epiglottis. Earlier you have seen the epiglottis. Epiglottis is a elastic cartilage which is covered by some mucous membrane. Anterior surface or the lingual surface of the epiglottis comes in contact with the food material, and it is lined by stratified squamous epithelium. The posterior surface, especially the upper part, also is lined by stratified squamous epithelium. The lower part is lined by pseudostratified ciliated columnar epithelium, and there are lots of glands. In the lower part, there may be some mucus and serous glands, and in the upper part, along with the columnar epithelium, there are certain goblet cells. This is the coronal section of this larynx, showing the vocal fold and the vestibular fold. The cavity in between the vocal and vestibular fold is called ventricle of the larynx. The vestibular fold is lined by stratified columnar epithelium with cilia, and the vocal fold, that is wear and tear or there is vibration, it is lined by stratified squamous epithelium, and there is no gland. Just below the stratified squamous epithelium is connective tissue. This is very important surgically because lots of infection occurs in this space. This has a fine name. This space has fine names. Below the vocal fold, the larynx is lined by respiratory epithelium, and there are certain glands, certain mucous glands and serous glands. The vocal fold contains some ligaments, some fibrous tissue, and certain muscles. The vocalis muscle is there. Those are the skeletal muscles. This is also showing the section of the half of the vocal fold. The upper fold is also called false vocal fold, and the lower fold is called the true vocal fold. You can see the lining epithelium, upper part is respiratory epithelium. The ventricle is also lined by respiratory epithelium, but the vocal fold is lined by squamous epithelium, and just below the epithelium there are lots of uh, lymphatic tissue and glands in both the false vocal fold and the true vocal fold, and deep to those folds you can see the muscles. Those are the skeletal muscles. The larynx continues as trachea, which bifurcates into two primary bronchi. On the right side, the primary bronchi divides into three to enter into the lung. There are three lobes in the right lung, and the left side there are two lobes. So the primary bronchi divides into secondary bronchi or the intrapulmonary bronchi. The intrapulmonary bronchi divides to form the segmental bronchi, and these are also called tertiary bronchi, and they supply the bronchopulmonary segments of the lung. There are 10 bronchopulmonary segments of the right lung, and 10 bronchopulmonary segments are there in the left lung. Then what is a lobule of a lung? This tertiary bronchi divides and subdivides up to 12 generations, and it comes up to 1 millimeter in diameter. A portion of the lung which is supplied by a bronchiole is called a lobule. So this is one of the lobules. And what is acinus? The bronchiole divides into respiratory bronchiole. A portion supplied by a respiratory bronchiole is called acinus. The acinus of lung. It is comparable to a glandular tissue. A gland is a lobule of a gland is formed by multiple acinus or acini, and uh, many of the acini joins to form a lobe. Similarly, in the lung there are lobe, lobule, and an acinus. Lobe is supplied by the primary bronchus, lobule is supplied by the bronchiole, and acinus is supplied by a respiratory bronchiole. This is showing the diagrammatic representation of the various divisions of the tracheobronchial tree. So first is segmental bronchus, then large segmental bronchi, small segmental bronchi, then comes the, after many subdivisions comes the bronchiole and the terminal bronchiole. The bronchiole has a lot of differences between the bronchi and bronchioles are there. See, the green colored area is a portion of the bronchiole which supplies group of acini, and one acini is supplied by the respiratory bronchiole. Trachea. Trachea is a tubular structure, and there are C-shaped cartilage which forms the skeleton of the trachea, and in the back this C, the ends of the C is joined by smooth muscles. The other structures of the trachea are: trachea has a mucosa which is lined by respiratory epithelium. It has a submucosa which contains the different loose connective tissue, and it also contains certain glands and uh, and certain lymphoid tissues also. The next structure is the cartilage, and beyond the cartilage is the adventitia containing the elastic tissue, and some connective tissue. Cell types lining the tracheobronchial tree. These are ciliated columnar epithelial cells, 30%. The goblet cells are there, 30%. There are basal cells, 30%. And there are other groups of cells which are present more on the terminal sides. These are the Clara cells, granule cells, brass cells, intermediate cells, and the serous cells. One by one, we will see the details of these cells. So, the whole tracheobronchial tree has linings and 8 types of cells has been identified. The main cells are the columnar cells, ciliated columnar cells, goblet cells, and the basal cells. The other smaller cells are the Clara cells, granule cells, brass cells, intermediate cells, and the serous cells. Here is a schematic diagram of the terminal part of the respiratory bronchial and where it is entering into the alveoli. In addition to the 8 types of cells, in the alveoli there are type 1 and type 2 alveolar cells are there, and macrophages are there which lies in the wall, sometimes in the cavity. The ciliated columnar epithelial cells, they are very tall cells, two low columnar epithelial cells, and they are covered by cilia. The cytoplasm contains numerous mitochondria near the cilia, and it has eukaryotic nucleus. It possesses 200 to 300 cilia per cell, and cilia also vary in length 4 to 6 micrometer. The cilia moves from the alveoli towards the oral cavity, and there is a movement called the mucociliary escalatory movement. What happens because of this movement? The mucus moves from the alveoli towards the oral cavity. The blunt-ended tips of the cilia bear a number of projecting filaments. They extend into low viscosity fluid, submucosal glands, but the tips are in contact with the thicker mucus that is secreted by the goblet cells. The rate of ciliary beating is 12 to 16 per second, and it is also called mucociliary rejection current, MCRC. Air pollution, smoking, infections, metaplasia, uh, these are the causes for damage of the cilia, and it produces several diseases which is called immotile cilia syndrome. If the cilia doesn't beat, what happens? There will be collection of the mucus which leads to infection and give rise to this immotile cilia syndrome. There are ciliopathies also, and certain genetic disorders are there with ciliopathy. The goblet cells are the second type of cells. They are present in the trachea and small bronchi. They are absent from the bronchioles. Actually, in the bronchiole, there are a lot of changes happen. So, one of the changes that up to small bronchi, there are goblet cells, but in the smaller divisions, that is the bronchial, goblet cells are absent, and the goblet cells are 6,000 to 7,000 per square millimeter of bronchus, and there are apical cyst, which contains dense vesicle containing the mucus. It is just like goblet cells of the GI tract, and in chronic irritation of the mucosa by dust or by smoking, this goblet cells increase in number, and a lot of mitotic activities are shown during infection or in the smokers. There are other cells called the serous cells in the lining of the respiratory tract. These cells are columnar cells, and they have irregular nucleus and with rough endoplasmic reticulum, supranuclear granules. They are the source of IgA in the bronchial mucosa, and they contribute to low viscosity circumciliary fluid. So serous cells also contribute in the secretion of the tracheobronchial tree. Clara cell needs a special mention. This Clara cells, the green color cells depicted here are the Clara cells. They are restricted mainly to the terminal and respiratory bronchioles. They are cuboidal, non-ciliated cells. Their apex bulges from the luminal surface. Numerous dense secretory granules, 500 to 600 micrometer lysosomes, are there in the apical part of the Clara cells. They are the important source of surfactant proteins. They have a functional similarities to the type 2 alveolar cells in the lung. The proposed functions of the Clara cells are: they secrete enzymes for breakdown of local mucus, protection against deleterious effects of inhaled toxins and carcinogens, a possible detoxification function like P450 enzyme is there, protection against emphysema. They have anti-protease function also. The secretion of the Clara cell has surfactant action. They secrete IgA, lysosome, cytokines, regulate the inflammatory response, and they are possible stem cells. After the damage of the bronchi, they help in the repair of the other cells. The Clara cell can give rise to other cells of the tracheobronchial tree. Dense core granule cells or granule cells, sometimes they are called T cells, Kulczynski cells, are neuroendocrine or apud cells. They are round to elliptical in shape. The basal part contains lot of granules. These are dense vesicles. They contain neurotransmitters like serotonin, calcitonin, generating peptide, somatostatin, bombesin, gastrin releasing factor. The functions of the dense core granule cells are regulation of bronchial secretion, smooth muscle contraction, and chemoreception. They discharge their granules in response to hypoxia. The brush cells. The brush cells are actually columnar cells, maybe cuboidal also. They have large, blunt, multivesicular at the apex. The base has a synaptic contact for gel sensation, and they are specialized for transduction of gel sensation. They are few in humans. There are intermediate cells, those are sometimes called basal cells also. They are present in the trachea and bronchi, columnar, and they generally lack cilia, dense cytoplasm lacking secretory granules. They are immature form of ciliated or secretory cells. The distribution of cell types. This grey color area, mostly it is the trachea and primary bronchi, where there are ciliated columnar epithelial cells, goblet cells, basal cells, then seromucous glands are there, neuroendocrine cells, and brass cells are there. The green portion is the secondary bronchi. They have ciliated columnar epithelium, basal cells, seromucous gland in the upper part, and brass cells. The pink colored area is the bronchiole. They contain ciliated and non-ciliated columnar epithelial cell, Clara cell, brass cells. The blue color area is the respiratory bronchial and the terminal bronchial. It contains ciliated and non-ciliated cuboidal epithelial cells, Clara cells, and type 1 pneumocytes already starts here because the alveoli are opening into these bronchioles. And this peach color or magenta color area is the alveoli. The alveoli are lined by type 1 pneumocytes and type 2 pneumocytes. Trachea. Already you have done the gross anatomy of the trachea. It has C-shaped cartilage. The diameter is 2.5 centimeter. Length is 10 centimeter. The function is conduit for air, and it conditions the inspired air. It has C-shaped hyaline cartilage. The ends of the C are joined by smooth muscles, and it divides into the principal bronchi, right and left principal bronchi. Right has 3 secondary bronchi, and left has 2 secondary bronchi, and the secondary bronchi divides into tertiary bronchi, which supplies the bronchopulmonary segments. The trachea has mucosa, then submucosa, the cartilaginous layer, and the adventitia. The mucosa is lined by pseudostratified ciliated columnar epithelium with goblet cells. The submucosa and the lamina propria are continuous. The boundary between the submucosa and the lamina propria is by some elastic fibers and cartilage. The submucosa contains lots of glands. Those are the mucus glands and serous glands, and it contains lot of lymphoid tissue, especially the plasma cells and the macrophages. Behind this is the cartilage. The cartilage ends are joined by muscles, and most outside there are adventitia. The adventitia contains elastic fibers and connective tissue fibers. Secretion of the goblet cells and the mucus and serous cell keeps the epithelial surface wet. This is the trachea, and this is the scanning electron microscope of the upper part of the cilia of the pseudostratified ciliated epithelium, and in between are the goblet cells. The goblet cell increases with chronic irritation like in smokers or in some infections, the goblet cells increase in number. The layers of the trachea are thick basement membrane and glossy homogeneous, densely packed collagen fibers. They become thicker in smokers. The lamina propria, which contains loose areolar tissue, cellular components are lymphocytes, plasma cells, eosinophil, fibroblasts, and lymphatic tissue, BALT. The boundary between the uh, mucosa and submucosa formed by elastic membranes. The submucosa contains lymphatics, loose areolar tissue. LAT is loose areolar tissue, submucosal glands, lymphatic tissue, large blood vessels, and these glands are more on the posterior side. The histological feature of the bronchus is similar to the trachea. It has mucosa which is lined by pseudostratified ciliated columnar epithelium. As the lumen decreases, the height of these cells also decreases. These cells lies on a basement membrane which is very much conspicuous in the primary bronchi, but in the lower division it slowly disappears. The lamina propria is similar to trachea, but the amount of areolar tissue becomes less as the diameter of the lumen decreases. It has a muscular mucosa also, contains uh, it contains uh, more attenuated and loosely organized connective tissue. In addition, there are some muscles uh, which lines the bronchial lumen completely. The smooth muscles become increasingly conspicuous in the lower part of the bronchial tree as the amount of cartilage decreases. It has a submucosa similar to the trachea. The cartilage layer in the trachea, cartilage was single, but here there are irregular cartilage. That's why the sections of cartilage are visible in the periphery of the bronchus. The next layer is the adventitia, which contains connective tissue, contains mainly the elastic fibers and collagen fibers. Here is the different divisions and subdivisions of the tracheobronchial tree up to the terminal bronchiole, respiratory bronchiole, and the ascending. The bronchus has the lining epithelium, pseudostratified ciliated columnar epithelium, surrounded by lamina propria and submucosa. Beyond that, there are cartilages, there is the hyaline cartilage, and in between there are smooth muscles. The bronchiole is the smaller division of the bronchi, and there are several changes occur in the bronchiole. Here, the cartilage is totally absent and the smooth muscle encircles the whole lumen, and because of the contraction of the smooth muscle, the lumen looks irregular, as noted in the arteriole also. Arteriolar wall, muscular artery, the muscles contract, that's why the lumen becomes irregular. Here also similar, but here it is the lining epithelium is ciliated columnar epithelium, which becomes only columnar and cuboidal. In lower divisions, it becomes cuboidal. So, this is a picture of the bronchiole or the terminal bronchiole, where the mucosa is folded and very conspicuous lining is the smooth muscle layer. Here also you can see the lumen, star-shaped lumen, epithelial lining, some mucosa, and total absence of cartilage. In the epithelium, some changes occur. The, the goblet cells are absent. So there is change in the epithelium also. It becomes columnar to cuboidal. Goblet cells are absent. Cartilages are absent. And it is replaced by smooth muscles. So epithelial height decreases. The goblet cell number also decreases. Subepithelial glands also decrease in number. Smooth muscle increases. Cartilage plates are absent. The large diameter bronchioles has pseudostratified ciliated columnar epithelium. The smaller diameter bronchioles has simple ciliated columnar epithelium, and the terminal bronchiole has simple cuboidal epithelium, and Clara cell starts at the bifurcation of the terminal bronchiole. Bronchial and bronchiolar muscle has special property. The total cross-sectional area of all bronchioles combined is far greater than that of the rest of the conducting passages combined. Thus, the tone of the bronchiolar smooth muscle efficiently controls the resistance to airflow with the lungs. These muscles are supplied by autonomic nerves and they are under control of hormones. They are supplied by sympathetic. Sympathetic stimulation actually increases the diameter, and parasympathetic stimulation decreases the diameter of the bronchiolar muscles. The terminal bronchiole. The terminal bronchiole has special types of cells are the Clara cells. The Clara cells actually project into the lumen of the bronchiole and they lie in between the ciliated columnar epithelial cells. The Clara cells contains RER at the basal part because they synthesize protein and these proteins are processed in the Golgi apparatus to form the the secretion or the covering of these secretions cover the Clara cells and the other cells and which has surfactant function. This is the Clara cell. The bunch of Clara cell surfaces look seen from above, and there is a small ciliated columnar epithelial cell. The Clara cells increase in number as ciliated cells decrease in number in bronchioles. They display in transmission electron microscopic characteristics of protein secreting cells. They secrete a surface active agent, a lipoprotein, that prevents luminal adhesion. Clara cell produce secretory protein, that is Clara cell secretory protein CC16, that increases in chronic obstructive lung diseases and in asthma. Respiratory bronchial, alveolar ducts and sacs. After the terminal bronchiole comes the respiratory bronchiole. Respiratory bronchiole name is given where respiration is possible. See, there are alveoli which opens into the bronchiole. So, to identify a respiratory bronchiole, you have to search the alveoli. So, The respiratory bronchiole is that part of the bronchiole where respiration is possible because the alveoli starts opening into the bronchiole. The other structures are similar. It has a mucous membrane which is lined by ciliated columnar, simple columnar or cuboidal epithelium. There is no muscle, there is no gland, Clara cells are there. This section shows the respiratory bronchiole. Here the alveoli are opening into the bronchiole. Why we are calling it the bronchiole? Because it has smooth muscle also and cuboidal epithelial lining. Lots of alveoli are opening into the respiratory bronchiole, which leads to the opening of the alveoli. So the whole thing here, what you see is the respiratory bronchiole lined by cuboidal epithelium. Outside is the smooth muscle. This is also showing the bronchiole in the upper part where you can see bits of cartilage. In the lower part there is no cartilage, and this is the terminal bronchiole, and terminal bronchiole continues into respiratory bronchiole where the alveoli are opening. In these two pictures also showing the opening of the alveoli into the bronchiole, and after the respiratory bronchiole the alveolar sac is there. Alveolar sac is a channel or opening where lot of alveoli opens into it. Here also one can see the alveoli part of the respiratory bronchiole because the alveoli are opening on in it. Terminal bronchiole, respiratory bronchiole, alveolar duct, and the air sacs are here. What happens in asthma? There is hypersensitive responses in the airway inflammation because of some dust comes, and this dust reacts with the epithelium, and there are lots of lymphatic tissues, and the lymphatic tissues actually that phagocyte the dust particles or bacteria or virus, and from the macrophages there are mast cells are there, plasma cells are there. There is liberation of different kinds of lytic enzymes. There are T cells, eosinophils. So these secretions irritate the glands in the mucosa of the bronchial tree, and the lumen gets obstructed. There is vasodilatation of the vessels also, and because of the increased vessel permeability, there are collection of fluid that reduces the lumen of the bronchiole, which causes. These are the picture of the alveoli. The mean total area of the alveoli is 80 to 140 square meter, the size of one tennis court. The total capillary surface area is 126 square meter. The total volume of the alveoli are 231 milliliter. There are number of alveoli maybe up to 150 to 300 million alveoli are there in both the lungs. Let us see the structure of the alveoli. The alveoli are lined by pneumocytes, the type 1 and type 2 pneumocytes. Here are alveolar cells. The type 2 pneumocytes project into the lumen of the alveoli, and type 1 alveoli are flat cells, the squamous cells. Type 2 are cuboidal epithelial cells, and there are lots of capillaries in the wall of the alveoli. Inside the alveoli, there are macrophages which are lying in the alveolar wall or in the alveoli itself. This is a schematic representation of the terminal part of the uh, bronchial tree, the respiratory portion, and the alveoli where air flow has been shown. Already I have mentioned there are two types of alveolar epithelial cells, the alveolar type 1 and type 2. Type 1 alveolar pneumocytes, they represent about 40% of all cell population of the alveoli, but they occupy 90% of the surface area. They are simple squamous cells. They have a central nucleus and highly attenuated cytoplasm, 0.5 to 0.2 micrometer. They have endocytic vesicles in the cytoplasm. Their perinuclear cytoplasm has mitochondria, little smooth endoplasmic reticulum, and occasional lysosomes. Gaseous exchange occurs through these type 1 cells. Actually, type 1 cells are the cytoplasm spreads throughout the alveoli, so they cover 90% of the alveolar wall. Broad air barrier. This is showing the broad air barrier. It is the type 1 epithelial cells or the cytoplasm of the type 1 epithelial cell. Then it lies in the basal lamina. Next is the basal lamina of the capillary and the cytoplasm of the endothelial cell. So the right side is showing a capillary. The capillary has endothelial cell. This is the cytoplasm of the endothelial cell, which lies on a basal lamina. Then on the other side there is cytoplasm of the type 1 alveolar cell, which also lies on the basal lamina. Sometimes the basal lamina, both the basal lamina are fused. Sometimes in between there are collagen fibers and elastic fibers are there. The nucleus is shown here. Most of the organelles lies around the nucleus. So there is a thin portion of the blood air barrier and thick portion of the blood air barrier. The exchange of gas occurs through the thin portion of the alveolar wall or the interalveolar wall. This is interalveolar septum, showing the thick portion and the thin portion. Thin portion is lined by type 1 epithelium, type 1 alveolar epithelium, which is squamous epithelium. Cytoplasm spreads to a large area. Then it lies on the basement membrane or basal lamina. Outside it lies the capillary. Capillary has an endothelium and a basement membrane. So this is a thin portion of the uh, interalveolar septum through which exchange of air occurs. Oxygen from the uh, alveolar air goes through this thin portion to the capillary, and from the capillary carbon dioxide comes out into the alveolar air. In the thick portion, if there is any fluid accumulation, it occurs in the thick portion of the alveolar wall. This is showing the representation of the interalveolar septum, which has the type 1 and type 2 pneumocytes, which lies in the basement membrane, and there are collagen fibers and elastic fiber proteoglycans. There are fixed and migratory cells, and free fluids are also there. And this is showing the capillary. Type 2 alveolar cells, or the pneumocytes, type 2 pneumocytes. They represent 60% of the alveolar wall population, but occupy only 5 to 10% of the alveolar surface area. So this is the type 2 epithelium. Number of epithelial cells are more than the type 1, but they occupy only 5 to 10% of the alveolar wall. The rest of the alveolar wall is occupied by the type 1 cells. They are rounded cells which are commonly located in obtuse angle, and they protrude from the alveolar surface. They have round ducts containing nucleus. The cytoplasm is rich in mitochondria, RER, ACR, lysosomes, and electron dense vesicles. They secrete surfactant. They decrease the alveolar surface tension and prevents collapse of alveoli. After injury of the lung, they divide by mitosis and they repair the lung alveoli. The most important function of the type 2 pneumocytes are secretion of surfactant, and the surfactant actually reduces the surface tension, that prevents the collapse of the alveoli during expiration. Without surfactant during expiration, the alveoli will collapse. This is representing the type 2 alveoli. It contains lots of lamellar bodies. Actually, these lamellar bodies contains lots of chemicals, that is the cholesterol, phospholipids, proteins, which forms the surfactant. The surfactant active agent reduces the alveolar surface tension. They prevent the collapse of the alveoli during expiration, and they facilitates expansion during inspiration. Already we have mentioned that there are lots of macrophages which lies inside the alveolar cavity or in the wall of the alveoli. The macrophages are formed from monocytes and some other hemopoietic tissues which lies in the alveolar septum, and they actually engulf a lot of dust particles like coal dust, asbestos dust, and they produce lot of disease due to irritation, chronic irritation by these dust particles. You have seen the surfaces of the lung look black. So the carbon particles which come through the inhaled air are engulfed by the macrophages, and these macrophages do not travel through the circulation. They lie in the lung alveolar wall, and that is why the lung looks black in citidolex. So this is showing the different portions and the composition of the different parts of the tracheobronchial tree. This is the conducting part and this is the respiratory part. The conducting part is up to the terminal bronchial and the respiratory part is starts from the respiratory bronchial and it ends at the alveolar. The conducting part contains all 8 types of cells. The main cells are pseudostratified ciliated columnar epithelium, goblet cells. These are the main cells of uh, the conducting part. So here in representing the epithelium, the pseudostratified ciliated columnar epithelium is there up to the segmental bronchus, and from terminal bronchus onwards, this epithelium becomes simple columnar, then cuboidal, then it becomes cuboidal and flat in the alveoli. Goblet cells are present up to the segmental bronchus. The ciliated cells are present up to the segmental bronchus and intermediate ciliated cells are present in the terminal bronchus. The glands are present all throughout up to the segmental bronchus. In the bronchiole, glands are not there. The cartilage are there up to the terminal bronchus. Cartilage are absent in the bronchiole. Smooth muscles are present all throughout up to the bronchiole. It becomes very thin in the respiratory bronchiole and it is absent in the alveoli. Elastic fibers are present all throughout. So, in summary, in this class we have studied the epiglottis. Epiglottis has an oral surface and inside there is a elastic cartilage. The oral surface is lined by stratified squamous epithelium. The lingual surface or the oral surface is towards the oral cavity. It comes in contact with the food material. The upper part of the posterior surface also is lined by the stratified squamous epithelium. The lower part of the epiglottis is lined by respiratory epithelium and there are mucous and serous glands in the submucosa of the epiglottis. The larynx has vestibular fold and vocal fold. Larynx is lined by pseudostratified ciliated columnar epithelium. The submucosa contains mucus and serous glands. There are two folds, the vestibular fold and the vocal fold. The vestibular fold is lined by respiratory epithelium and lots of glands are there. The space between the vestibular and vocal fold is called the ventricle. The vocal fold, as because there are a lot of vibrations, it is lined by squamous epithelium, deep to which lies the vocalis muscle. The larynx continues as trachea, which has C-shaped hyaline cartilage in the center, and in the lumen is lined by respiratory epithelium, that is pseudostratified ciliated columnar epithelium with goblet cells. The submucosa contains a lot of mucus and serous gland. The cartilage is connected posteriorly by smooth muscle, and its adventitia outside the cartilage there are elastic tissue and reticular tissue. The next division is the primary bronchi. The structure is like the trachea, but it has irregular cartilages. That's why sections of cartilage will be seen in the third layer. Otherwise, it has a mucosa lined by cytostatic ciliated columnar epithelium, submucosa containing glands, and third layer is formed by the cartilages. Fourth layer is adventitious. Next comes the bronchiole. The most conspicuous feature in the bronchiole is the absence of cartilage. Please remember, most conspicuous feature is the absence of cartilage. Instead, there are smooth muscles. And because of the contraction of the smooth muscles, lumen becomes star-shaped or irregular. The mucosa is lined by pseudostratified ciliated columnar epithelium in the upper part. The lower part is lined by simple columnar to tuberal epithelium. The goblet cells are absent in the mucosa. The respiratory bronchiole has all the features of a terminal bronchiole, and the alveoli open into the lumen of the respiratory bronchiole. The alveoli contains two types of cells, type 1 pneumocytes, which are nearly 40% of the cell lining the alveoli, and 60% cells are type 2 pneumocytes. Type 1 pneumocytes, through which the exchange of gases continues or occurs. Blood-air barrier is formed by the cytoplasm of the type 1 pneumocyte, basement membrane of the pneumocyte, basement membrane of the capillary, and the endothelium of the capillary wall. They form the blood-air barrier. The type 2 pneumocytes contain surfactant. They secrete surfactant which reduces the surface tension, thereby it prevents collapse of the alveoli. The surfactants are produced after 35th week of gestation. So if a baby is born before 35th week, they suffer from respiratory problems and they need respiratory support for their survival. In some babies, in premature babies, the surfactants are not produced, and it produces a hyaline type of membrane in the alveoli, and that is called hyaline membrane disease. Thank you all.